Good morning, everybody. My name's Lorelle Sherman. I'm with Oregon State University Extension, and I invite you to come sit in your seats.
Hello.
Hi. Nice to meet you.
Nice to meet you. Thanks for moderating.
Absolutely. All right, so let's get started. Our first session is Disturbance in Early Seral Ecosystems. It's timely that I'm moderating for this session because in just a couple of weeks, we're going to have 3 interpretive signs highlighting early seral forest habitat placed in the McDonald Dunn Research Forest. So if you're local, I encourage you to go check those out. So, our first speaker today is Lauren Ponisio, and they are here to talk about enhancing floral resources for pollinators in harvested forests. Please, please welcome me in joining Lauren.
Thank you. Good morning, everyone. I'm Lauren Ponisio. I run a lab at the University of Oregon. We do some ecology. But it's lovely to be up here in Corvallis. Our lab focuses on bee community ecology and particularly restoring bees in agriculture and now, since moving to Oregon, harvested forest landscapes. So pollinators are awesome. They pollinate about 88% of flowering species and all the best things in life, like blueberries, chocolate, and coffee. Oregon is actually home to about 700 and counting species of bees. And I usually talk about bees when I talk about pollinators because they are generally the most dominant and efficient pollinators. So if you go into any ecosystem, it's probably the bees doing most of the work. However, sadly, a lot of bees are declining. I'm sure you have all heard about it. And in Oregon, we have quite a few, and particularly these big 4 bumblebees that are either going to be petitioned to be federally listed or gaining Oregon special status. And so some of them have really strange names like the obscure or fog belt bumblebee. But of particular relevance is the western bumblebee, which used to be the most common bumblebee across the entire West. And its populations have declined by over 90%. What is causing this? A big part of it is just the loss of florally rich habitats. And if you are, research starved, then you're probably going to end up with being affected by other stressors like disease. And so we want to ask, how do we support more florally rich habitats in Oregon to support healthy pollinators? And one place that pollinators love to be is potentially in harvested forest sands. So, you remove all those pesky trees and you end up with this beautiful, early seral habitat. There have been some studies done in harvested forests showing that, we have these different stands, they have uniform ages, they're in this rotational system. This creates a really lovely mosaic for pollinators and other wildlife, if you care about those. So, we have these nice early, like when there's all these flowers or all these bees. And this lasts, at least in the Coast Range, in about years 1 to 6. We probably have a little more time in the Cascades where there's less water. And so we have this really nice peak, and then, unfortunately, the trees grow. But luckily, then they harvest them, and it starts all over again. But so some private forestry practices, and particularly wildfire, can influence the landscape. This is during the winter. So there's not as many flowers out there, but this is like kind of double compounded disturbance of timber harvest and wildfire can promote the growth of disturbance resilient and invasive species. That can then lead to needing to selectively herbicide, which can reduce overall floral availability. And unknowingly, for this really nice rotational mosaic, if you have a really large wildfire and everything gets harvested, that ruins that kind of mosaic pattern, though it's very convenient for experimental design. So we want to ask, can we boost pollinator communities by seeding native wildflowers into these salvage log stands? We did an experiment funded by the USDA out in the Holiday Farm Fire. We had 3 different landowners, 2 large and 1 small. And we looked at areas where we could have these, enhancements of wildflowers next to controls. And this was distributed across the wildfire. Particularly what we did is we seeded into burned slash piles. So we'd done a previous experiment where we found that if we planted native seeds into these burned slash piles, they did a lot better than if we planted directly into the harvested stands. And so there's some little circles of all these little burn piles out there. And so, those were, of course, previously burned. And then we went ahead, and so this is what they looked like before seeding, and then 1 year post seeding, and 2 years post seeding. And they continued on. We've now surveyed to 3 years post seeding. And in general, you can see that native wildflowers established pretty well, but I'll show you some data on that. So particularly what we saw is as you went from year 0, so those are the controls, or the year before a burn/pile was seeded, all the way to year 3, we saw a higher probability of just having flowers as time went on. So a little under 50% of the controls, those non-seeded burn/piles, never had any flowers in them at all in this 4-year period. And we started sampling in 2021, so the year after the holiday farm fire. However, when we look at the actual number of flowers and the floral diversity, so this is like given you have flowers, what's the abundance of flowers? So this is the number of flowers, and what's the diversity? And these are pretty small plots, so they're 2 meter by 2 meter plots. We don't really see much change until about year 3. And then my grad student and I had to have, the talk, the statistical significance versus biological significance talk, where we really see a pretty stable floral abundance and diversity. And so ignoring the controls, this is actually a good thing when you're thinking about restoration. This means, we seed things in there, and in general, the plants, they keep blooming year after year after year. We're not seeing big declines in floral abundance or diversity. However, we were really expecting there to be more flowers than in the controls. And so, but we're examining this a little bit more. And what we think is going on is, even though, let's say, the places without flowers ever, and there are a lot of them, but if you did have flowers, those controls were actually pretty nice looking. A lot of them were right next to riparian buffers. They got colonized by a lot of different species. Some of those species were, fireweed and false dandelion, but bees do like those. So we're looking more into the composition of these different communities. Next, we wanted to look at what about pollinator abundance? How does that change with years post-restoration? And what we saw is that, if you didn't have any flowers, unsurprisingly, you didn't have any pollinators. And so the probability of having pollinators increased with increasing floral abundance. And then we saw some differences with this year's post-restoration. So it's like we're kind of gradually increasing the number of bees we're catching in these plots within a 15-minute period, again, in a 2 meter by 2 meter plot. So there's just not that many bees. But by year 3, we kind of see this little drop off again. One pickle with our experimental design is we're also impacted by years post-fire. So there is also this annual variability of how good those years were. And so we're further looking into that, but as you can imagine, we can't control everything. So in terms of, we do see a strong effect of restoration on the probability of having flowers and pollinators, but much weaker effects on the abundance and diversity. So in general, did they work? And I'm just going to say this is a bit TBD because right now we're just analyzing the data on those really small plots, but we're gonna dig a lot deeper in the next year. So we have almost 3,000 bees screened for parasites. We still don't have some of our species IDs back from our 2 taxonomists. I text them once a week. So I'm very jealous about all the people who, come back from the field and know their species IDs, but we're still waiting. So what we'd love to do is to analyze, both the real landscape level effects and not just those tiny little plots, but how these are affecting the entire stand, as well, and incorporating landscape level variables like those different sizes of fire, severity fire, and analyzing more the composition of species in these different places in different functional diversity metrics. So right now I'm just piling all pollinators into a bin. They're all very different. And so we'd like to really look at the differences in their traits and life histories. And with that, many, many people have been part of this, including my lab, all the wonderful people at Cassie. We are collaborators with Jim Rivers at OSU and his grad student Lily, our wonderful taxonomists who, they love being texted by me once a week. I'm sure it's gonna be great. And all the landowners who let us do these plantings on their land. And we are holding questions until the very end. Is that correct? Yes. So thank you very much. And I'll pass it to the next person.
Thank you, Lauren. Yeah, we will have time at the end, hopefully for questions. So please scan that QR code if you'd like to submit a question. Next, we have Claire Goodfellow, who's going to be talking to us about effects of deer forage in the Tongass National Forest. And let's see if I can get it right this time. Please join me in welcoming Claire.
Okay. Yeah. Hi, everyone. I am Claire Goodfellow. I'm a research associate in Tall Levy's lab at Oregon State University. And you may have noticed my talk today is going to take us a little bit afield of the state of Oregon. But I'm hoping that by the end of it, I'll have demonstrated kind of the utility of these new genetic methods as a monitoring tool for evaluating the impacts of forest management practices on wildlife. So the work I'm talking about today is focused on the Tongass National Forest. This is America's largest national forest. It's about 17 million acres, which also makes it the largest intact temperate rainforest in the world. And to contextualize its sheer size, you can consider that the Tongass is larger than all of our national forests here in Oregon put together. And as a national forest, this makes this absolutely massive tract of land a multi-use landscape that must be jointly managed for both timber production and many wildlife species, including the endemic Alexander archipelago wolf, all 5 of our Pacific salmon, and the star of our show today, the Sitka black-tailed deer. These deer are endemic to the coastal rainforests of northern British Columbia and southeast Alaska, and they're both an important subsistence resource for regional communities and a key prey resource for those endemic Alexander archipelago wolves. And in particular, it's the Tongass's mandate to jointly manage for both these culturally and ecologically important deer and economically important timber resources that really it's the underlying motivation for our work. So to delve into the deer-timber relationship a little deeper, I've included this conceptual diagram of deer carrying capacity in the Tongass along a gradient of stand age. So starting on the left of the diagram, you can see that old-growth forest can support a large number of deer. If that forest is clear-cut, the shading effect from the overstory is removed and available forage spikes. But that increase is temporary as young growth canopy begins to shade out forbs and shrubs after about 20 to 30 years during the stem exclusion phase. And you can see that in the photo on the right. And this is then followed by several hundred years of natural succession when that understory forage for deer remains limited. And for this reason, the Forest Service is particularly interested in actively managing the 450,000 acres of young growth on the Tongass to improve forage for both deer and other wildlife that use the forest. And to date, pre-commercial thinning has really been the silvicultural intervention of choice. So about 200,000 of those 450,000 acres of young growth have been treated, with one of the key goals here being to boost forage for Sitka black-tailed deer in those young stands. And to assess the impacts of these treatments on deer, a series of experimental plots were established in 2001 as part of the Tongass-wide young growth studies. I'll just call it TWIGS. These experiments paired various thinning treatments with controls to quantify the impacts of pre-commercial thinning implemented at 3 different time points. Post-stand initiation, so 15 to 25 years, 25 to 35 years, or greater than 35 years after initiation. And data from these plots have shown that pre-commercial thinning does significantly increase understory biomass relative to control plots, regardless of that time point at which the thinning occurred. But what remains unclear is whether that additional biomass that's being generated by these thinning treatments are plants that deer actually like to eat. So in other words, are we just generating a lot of unpalatable, unpreferred forage, or are these treatments actually increasing the forage available to deer on the landscape? So to address this, I'm talking about kind of 2 separate questions today. The first one is, what do these deer actually like to eat? And the second one is, how can we manage the forest's young growth to improve the forage for the species? So on that first question, I used a method called DNA metabarcoding to quantify deer diet throughout the region. And this method works by using what are called universal primers to target and amplify a small portion of DNA that's considered to be hypervariable across different plant species. So here you can see that when I align these different species, Seral, red cedar, salmonberry, no two species look the same at that hypervariable region. And this essentially then enables us to use next generation sequencing to read this region like a standard UPC barcode and to subsequently identify which species each sequence in a mixed sample like a deer pellet is derived from. So diving into some results, today I'll be talking about a data set of 435 deer pellets that were collected from 2022 to 2025 in association with 24 of those twig sites. And in total, we detected 89 unique diet items. Between 1 and 18 different plant species were found per sample with an average of about 9 different plant species. About half of our samples were collected from April through May, which represents spring diet. On Prince of Wales, this time period is still considered kind of a pinch point, where snowmelt and seasonal greenup are just beginning to occur. So the plants that are consumed at this time might be considered more so fallback foods, not necessarily preferred, but seasonally important to getting the deer through the last of that harsh winter. So the highest frequency diet item in our spring data set was Western red cedar, which accounted for around 21% of our deer's diets. That was followed by vaccinium at 19%, bunchberry dogwood at 16%, and then rushes at around 9% of that spring diet. The other half of our samples were collected from June to August, which make up our summer data set. And compared to spring, this time period is more a time of forage abundance, where the diet might be more representative of deer's preference versus just what's available to them. And vaccinium comes in as the most consumed taxon in the summer at around 22% of the diet. That's followed by salmonberry at 21%. Again, red cedar, but now at 14%. And then finally, foam flower makes up 7%. And looking at these seasonal shifts, you can see a huge shift towards salmonberry in the summer. So this accounted for only around 2% of spring diet, but over 20% of the summer diet. And you can compare that to rushes, which are around 9% of spring diet, but virtually non-existent in the summer diets. And similarly, evergreen ferns like deer fern and licorice fern together made up around 7.5% of spring diet, but less than 1% of summer. And after looking at these seasonal trends, I then took advantage of the fact that these samples were collected from those twig sites, which all have high quality understory biomass data available. And I used selectivity analyses to look at how deer were using these plants compared to their availability on the landscape. So were they selecting for them, were they avoiding them, or were they just using them in proportion to their availability? And I stuck a vertical line here to show that everything to the left of the line is avoided, and the further left you go on this figure, the stronger the avoidance. So what really stands out here is that all 4 of the common fern species on the landscape came out as the most strongly avoided species by deer. And on the right-hand side of the figure, these are not plants that are selected for, but rather plants that are eaten in proportion to their availability. So this included vaccinium, which remember is the most consumed plant in the summer, but it is also the most prevalent plant on the landscape. And bunchberry dogwood also fell into that neutral category. In terms of what the deer are selecting for in this bucket, we have, amongst others, red cedar, alder, maybe unsurprisingly, deer cabbage, yellow cedar, and then brunettes. So at this point, I have a pretty good idea of what these deer prefer to eat. And in my last minute or two here, I'm hoping to pull in that second component, which is how can we manage the forest, the young growth in the system to improve forage for the species. So recall from earlier that pre-commercial thinning increases understory biomass in the Tongass regardless of the time point at which that thinning treatment is implemented. And the metric that was used in that work is total forage biomass, which is simply all of the plant biomass available to a deer on the landscape. But as I hopefully just demonstrated, deer don't necessarily use all of that forage available to them at a site, and forage metrics that account for deer's preferences would give more biologically relevant insight into whether different forest management techniques are in fact improving forage for these animals. So there are a few different ways to define biologically relevant forage, but today I will focus on accepted forage biomass, which are those plants that deer choose to eat at rates greater than or equal to their availability. So in other words, I'm just excluding the plants that deer are actively avoiding. And in our system, accepted forage cumulatively accounted for around 45% of total biomass. And we found that accepted forage was significantly higher in thinned plots than in neighboring unthinned controls. And that age at treatment also had a significant effect, with those plots that were treated at the 15 to 25 year time point having about twice as much accepted forage biomass as those stands that were thinned greater than 35 years. And this last point is notable as these results are consistent with work that came out earlier this year, which found that although total biomass increases across the board with thinning, binary browse probability, so the presence or absence of deer browse sign, is highest in plots that were thinned at those earlier time points, with plots thinned later not experiencing those same gains. So taken together, although the impacts of thinning are, of course, both system and organism specific, all of this work suggests that in the Tongass National Forest, pre-commercial thinning is an effective management tool to increase deer's forage on the landscape, especially if you implement it early. And then secondly, I hope that I demonstrated the power of DNA metabarcoding as a monitoring tool to evaluate the impacts of forest management practices on wildlife. Thank you to my collaborators and the folks who funded this work, the Forest Service and the Alaska Department of Fish and Game. Thank you.
Excellent. Thank you, Claire. Next up, we have Aidan Place from Oregon State University speaking about early seral bird communities in intensively managed forests.
Okay. Hello, everyone. My name is Aidan Place. I'm a first-year master's student at Oregon State University in Matt Betts's lab. I'm going to be talking today about the project I'm working on, which is assessing the long-term responses of avian communities to intensive forest management. So, long-term monitoring projects of bird populations in the Pacific Northwest have revealed some pretty worrying trends. I think we're all probably aware of the ongoing declines in northern spotted owls and marbled murrelets, both of which are species associated with old-growth forests. I think a bit less well known is that species associated with our youngest age class of forests, which is of course early seral forests, are also declining as well. So this is breeding bird survey data for two species, orange crown warbler and Rufous hummingbird, both of which are associated with early seral forests. And as you can see, there's been a pretty dramatic decline since about the mid-20th century in both of these species. This decline has been shown to be linked to losses in the amount of early seral forests available in the Pacific Northwest. This might seem initially kind of surprising. I suspect a lot of us have spent time in the Coast Range, or at least driven from here in Corvallis out to the coast. We've all seen the clear cuts out there. You would assume that this amount of forestry is producing a constant cycle of disturbance, creating new young forests across the landscape in the Pacific Northwest. This is both true and not true. In recent decades, industrial forest lands have undergone intensification, largely through the use of herbicide. Herbicide works to reduce competition for resources with planted tree saplings by reducing other vegetation during the first few years of stand growth. And in doing so, it produces a less diverse and more homogeneous forest. This seems like a pretty intuitive explanation for bird declines. There sure maybe is more young forest on the landscape, but it also may not be the type of high quality habitat that is sought out by birds. So to assess to what extent this was true, a study was set up in 2010 on private and state forestry lands in the Coast Range. The goal of this was to assess the response of bird abundance and diversity to a gradient of herbicide applications using a randomized block experiment. So there's 8 clusters of 4 stands, and then 1 of 4 herbicide treatments was applied to each stand in a block. So we have a control treatment, a light, a moderate, and an intensive. And as the slide shows, the intensive treatment is outside the typical operational range. And here's a sort of clear representation of the study area. So it's all throughout the Coast Range, and you can see there's 8 clusters of 4 stands. And zooming in on our Trask block here, you can see that there's 4 stands per cluster, each of which has received either a light, a moderate, a control, or an intense treatment of herbicide. And then within each stand, there's 3 survey points, which were surveyed for birds during the peak of the breeding season in June. And it should be noted that this surveying has happened every year except 2 since 2011. So these sorts of long-term experimental designs are really quite rare in ecology, which I think makes this study really exciting. And so I'll quickly go over some of the results of this study to date. So this is before my time of joining it. You can see here broadleaf plant cover by treatment, and you'll notice that indeed treatment intensity predictably had pretty dramatic effects on broadleaf cover. I think what's really fascinating here is that even by year 10 of the experiment in 2020, you don't see recovery, quote unquote, of broadleaf cover compared to the light and control treatments. And this illustrates how long-term the effects of herbicide can be on a landscape. And here's a visualization of what these look like on the ground and the differences in plant cover. This was on year 6 of the experiment, so soon after the cessation of herbicide application. And you can see that there are pretty dramatic differences in both broadleaf cover and vegetation cover on the whole between our control plots and our intensive plots. And so what about birds? How do they respond? Here's overall species richness compared to treatment. As you can see, there's indeed a decline in richness across treatments. You'll note that the response was strongest in years 2 and 3 of the study, which is kind of logical. That's in the thick of things as far as herbicide application goes. And kind of interesting is that the response has bounced back by about year 10 of the study. And as we saw a couple slides ago, that is asynchronous or decoupled from the response of broadleaf vegetation, where broadleaf vegetation is not on the same level across treatments by 2020, but it is more or less pretty similar, but in terms of species richness. And then broken down by guild, for the non-bird people in the crowd, leaf gleaners are species that feed on invertebrates on the margin of vegetation. It was hypothesized that these would respond pretty strongly to treatment because, of course, they require broadleaf vegetation for foraging. And this is indeed the case. Here's the leaf gleaners compared to, compared to non-leaf gleaners. And you can see that there are very dramatic differences. I think the results from the first few years of the study with leaf gleaning species is one of the more dramatic and interesting results that has come out of this to date. All right, so where do we go now with this? We're 15 years into the study. What can we gain from continuing to monitor it? So here's those photos again from 2017. And here's more or less what the stands look like now. So if you think about the original rationale for this work, the goal of herbicide application is to maximize efficiency by reducing competition, right? So intuitively, that truncates the early seral period on the front end by slowing the development of complex broadleaf vegetation. However, because increased growth efficiency should lead to earlier canopy closure, intensive forestry likely truncates the early seral period on the back end as well. So now, 15 years on, it's the second part of this sort of dual shortening of the early seral period that we're measuring. How are the effects of these differential herbicide treatments still being felt 15 years on via changes in the timing of canopy closure? And will the period of use for early seral-associated species be extended in stands that received less of a herbicide application? And another sort of thing we're continuing to look at this is a chronosequence study of bird responses to intensive forestry. So this is from a separate project. But as you can see, most species showed a unimodal response with a strong peak. And for a lot of species, that peak isn't until year 15, year 20 following harvest. And so in other words, even with our long-term 15-year data set, we haven't captured peak abundance for a lot of species yet. And so by continuing to collect this data in the experimental stands, we'll better capture the full ranges of species responses. And then the other thing that I'm really excited about that we're doing moving forward with this project is bioacoustics. So starting this past summer, I deployed autonomous recording units, or ARUs, which is essentially just a microphone, at every bird census point. And these recorded throughout the summer, allowing us to capture significantly more data than we could with just point counts alone. And the data from these ARUs can be downloaded. It can be transformed into a spectrogram image like you see on the right there. And then using machine learning automatic image classification methods known as convolutional neural networks, these can be automatically identified to species. And I think this new data is going to open up a lot of really exciting opportunities and avenues for research with this data set moving forward. So I'll just finish. I hope the audio works, but I'll just finish with a short snippet of the acoustic data that we collected this summer. And while it's playing, try to sort of imagine yourself as a field technician at sunrise on a really crisp summer morning in the Coast Range. And also imagine trying to sort of desperately take down point count data on all the species. So maybe, I don't know how this works, but is it possible to play the audio? I don't know if I can do that from here. Oh, I can. Oops. Okay, well, you can kind of hear it. Yeah, as you can see, these stands are pretty surprisingly biodiverse. And it's Jake Friscyle who made that little animation, so thanks to him for that. So yeah, that's all I've got. Thank you guys for listening.
All right, so we have time for 2 questions, and I know Aidan just spoke, so the questions haven't rolled in yet. So I'm going to ask Lauren and Claire to come up here to answer 2 questions, one each. So Claire, while you're walking up here, the question is, how did you quantify available forage on the landscape to the species level?
The available forage?
Yes.
Yeah, so the answer to that is I personally did not. This was a massive data set that was taken by the Forest Service. It's a 20-year data set at this point. And they had, I don't even know how many techs out at that point quantifying the biomass. There's a paper I could point you to, whoever asked that question, with the equation that they used to actually get the availability. But yeah, the Forest Service gave me the availability data.
And Lauren, there's a couple of questions here about what types of flower species did you see do best, planting timing, and I also see we have lots of woodland owners in the audience, so I think that might be a pertinent question to answer.
Can you bring up my slides? I actually have a really nice slide on this one. Is that possible? The very last slide of my presentation. We moved on. Okay, well, so we chose a mix of annuals and perennials. And we were very lucky because H.G. Andrews, the experimental forest, is just up the road. And we were able to get lists of early cereal species just naturally found in those forests. We then excluded all the non-flowering and invasive species, and also species that might be problematic for forestry or really likely to occur already, like fireweed. No one needs to plant fireweed. And then we, use the very technical kind of like bee senses, and we're like, we think bees would like that, as well as, so the very last slide, we are really lucky in this state to have the Oregon Bee Atlas, and they have been collecting data on data on like every flower that bees visit. And so we cross-checked it with those. And then we looked for seeds that were commercially available. And so in these stands, we have these mixes of 8 species each. If I were planting my own burn pile, I might go up a little bit higher. I might say like 15, 20 species. And we did a very technical thing. We mixed them with vermiculi, and then we Johnny Appleseeded them. And it went really, really well. We did small scale experiments with this and had incredibly high germination and flowering. And the reason why we did these little mixes is we were working with some plant people, and we were interested in how different combinations of species with a really wide range of pollinators and species with a more narrow range of pollinators might promote each other. But if I was a woodland owner, I would just take a whole bunch of different seed species that were found in my region that we could buy commercially and just Johnny Appleseed them. There may be some burn piles that you probably don't need to seed. This is kind of what we've been learning, like especially if they're right next to a riparian buffer or a source of a lot of native species. You may, but you're not going to do any harm. You'll just be potentially wasting a bit of money on seeds. So that's yes. And also very conveniently, OFRI has a lovely publication on enhancing bees in a harvested forest that has sources of commercial seed producers and recommendations on seed mixes as well.